In the final video in our series looking at digestive regulation, we're going to look at the functions of the final regulatory hormone, a hormone called cholecystokinin. Now, if you don't want to say cholecystokinin, instead you can remember three of the consonants in the word and use the short form CCK. Now, CCK is a critical hormone for regulating digestion in the duodenum, which not only is the primary target of the effects of CCK, but also is where cells produce CCK in order to cause its specific effects. And as we will see in this video, CCK is an integral hormone for regulating mainly the digestion of fat, but also the digestion of proteins in the duodenum. And again, fat is uniquely difficult among nutrients because fat is hydrophobic, meaning that it does not dissolve in water, which means that CCK is primarily going to affect the pancreas because the pancreas produces the digestive enzymes required to break down fats and all of the other undigested food, but it's also going to affect the liver because of one of the products that the liver produces in order to make fat digestion easier. Now, if we back up and look at the environmental conditions that trigger the duodenum to actually produce CCK in the first place, the first and strongest stimulus is the presence of fats detected by the cells of the duodenum. And of course, the main reason why this is such a strong stimulus is because up until this point, fats have not been digested at all. Remember that starch is the primary target of your saliva's digestive enzymes, and protein is the primary target of the digestive enzymes of the stomach, meaning that this is the first point in the digestive system where fats can actually be broken down. Now, the second stimulus, in addition to the detection of fat in the duodenum, is the parasympathetic response of your brain, which is again detected whenever there is food that has entered the digestive system, which is why we also call the parasympathetic response the rest and digest response. Now, in addition to telling the cells of your duodenum to produce CCK because your brain recognizes that food is on the way, it also is going to stimulate the liver to do what the liver is required to do in order to aid with fat digestion, and that's actually what we're going to start off with here. Now, the liver is a critical organ of fat digestion because it is the source of bile, which is the hydrophobic solvent that is needed to dissolve any fat and any other hydrophobic nutrients in your duodenum, because fat, of course, does not dissolve in water. However, remember that bile, although produced in the liver, is normally not transported to the duodenum directly, but instead is stored in your gallbladder until it is needed. So if there is any bile in the gallbladder, CCK is also going to trigger the muscles surrounding the gallbladder to contract. And when the gallbladder contracts, it squeezes all of the bile that is stored in the gallbladder into the duodenum. And when bile mixes with fat, because bile is an amphipathic solvent, that means that it is able to mix with both hydrophobic substances like fat and with water-soluble substances, allowing bile to dissolve fat, therefore making it easier for the pancreas to fulfill the next half of fat digestion. So the other primary target of CCK, of course, is the pancreas, because the pancreas is the organ that actually produces the primary digestive enzymes that are required in order for fat, as well as every other piece of undigested food, to be broken down within your duodenum. Now, as mentioned previously in our video on secretin, insulin is also quite important in the function of the pancreas because insulin regulates the absorption of glucose, and given that one of the digestive enzymes released by the pancreas is amylase that breaks down starch, that means that the the jejunum and the ileum, the subsequent sections of the small intestine, are going to be absorbing glucose, and therefore CCK also triggering the production of insulin allows the storage organs like the liver to absorb glucose from the blood that has been digested and store it for future use. So the point is that CCK allows the liver and the pancreas to work together in order to dissolve any nutrients that do not dissolve in water, so that the pancreas can use its digestive enzymes in order to break down any undigested food that makes it into the duodenum. And with the effects of CCK, this basically means that all food that is able to be digested by your enzymes will be broken down by the time they pass through the end of the duodenum. Now, the final fact that we need to talk about is how CCK is regulated and how it can be shut off, because just like the stomach, we do not want the presence of overactive digestive enzymes, because just like your stomach, the digestive enzymes from the pancreas will just as easily break down the cells of the duodenum if there is no food to digest, and therefore we need to look at how to shut off CCK release. So CCK release is actually relatively complicatedly regulated. The cells in the duodenum do not directly release CCK into the blood. Instead, when fat is detected by the duodenum, it triggers specific cells in the duodenum to release a protein called CCK release protein. And we will henceforth know CCK release protein as CCK-RP. So again, if we follow what CCK-RP does, when fat is detected by the duodenum, these tell the cells of your duodenum to produce and release CCK release protein, which then will tell these specific cells that produce CCK to actually produce CCK and release it into the blood. Suffice to say, if this release protein is not released, CCK will not be released into the blood and will never make it to the liver or to the pancreas. Now, one of the things that CCK does for the pancreas is it produces trypsin, which is a protein digesting enzyme. Now, as we've established, CCK release protein is a protein. So because of the fact that CCK directly triggers the production of trypsin, and because trypsin digests protein, trypsin, when it is produced, can actually break down CCK release protein, because again, CCK release protein is a protein and trypsin is an enzyme that breaks down protein. And if this release protein is not present, therefore this will inhibit the release of CCK, because if this release protein is not present, it cannot tell the cells that actually produce CCK to release it into the bloodstream. So as we can see here, the more fatty acids enter your duodenum, the more likely this release protein will be released and trigger the release of CCK. The more CCK is present, the more likely it will tell the pancreas to produce trypsin, and the more likely in turn that trypsin will digest this release protein and prevent CCK from entering the bloodstream. So this is yet another example of feedback inhibition, where let me just finish spelling inhibition properly, where the more the stimulus is active, the more likely the stimulus will shut down the results of that stimulus, which is exactly what feedback inhibition is, and this prevents overactivity of the pancreas and prevents the digestive enzymes of the pancreas from doing permanent damage to the cells inside of the intestines. This concludes our series on the regulation of the digestive hormones and the individual hormones themselves. To test your knowledge of the regulation of the digestive systems, you can take a look at this case study, which is actually investigating a disorder involving regulation of the stomach, specifically involving the regulation using gastrin. And after that, you can take a look at all of the practice problems below to test your knowledge of how well you know the effects and causes of each of the digestive hormones.